All right. So, hello and welcome today to our Kentucky Embrace seminar series. So, today we have two outstanding researchers from Eastern Kentucky University, Dr. Lisa Middleton and Dr. Laura Rowe. And we will start with Dr. Lisa Middleton, who is an assistant professor in the Department of Biological Sciences. And she will be talking about developing a cell culture model to examine signaling pathways altered by 1-methylpsilocin during embryonic development. So, Dr. Middleton, take it away.
All right, thank you. So I just want to first kind of give you all some background on how kind of the past year and a half of research has been going for me, and the fact that I've really kind of gone a couple of separate ways. First of all, I've really kind of allowed a couple of my undergraduates to take the lead on some of their own projects of particular interest to them. For example, I have a pre-physician assistant student who is interested in dermatology and skin cancer diagnoses and predictors of skin cancer. And so he actually took the lead to run a project on his own. And I've been there to kind of support him along the way. And second, I've also really been focusing in the past year and a half or so on collaborations with other faculty. That is one of the best ways around here for us to really make progress in the lab and perform experiments. And so I've worked at EKU with Dr. Lindsay Cormier, who is developing new medications as potential therapeutics for reproductive cancers. But the one that I'm going to talk about today is a collaboration that I have been starting, and this is in the early stages, with Dr. Max Azine from Loyola Marymount University in LA. And so I was approached by Dr. Azine at Loyola because of her interest in substance abuse, but the fact that it was a new area for her. And so I have worked with her on developing paperwork and submitting paperwork so that she can actually have illicit drugs at Loyola Marymount and approved by the state of California, and is now getting the DEA licensure for that as well, so that she can get that from the federal government. And so she really began the idea, of talking about psilocybin and the effects of psilocybin on embryological development. And I'll kind of give you a background of what I mean by that here. So first of all, in case you weren't aware, psilocybin is a drug that is found in the psilocybe mushrooms, and it is the main psychoactive component, and that main psychoactive function is to elicit hallucinations. Historically, it's really been used in indigenous populations in both South and Central America, and it's been used for religious practices. Recently, though, there has been kind of a shift in thinking about this drug as a potential therapeutic. And this was really the seminal work of Roland Griffiths out at Johns Hopkins University. And so when we talk about its potential as a therapeutic with regard to psilocybin, you can see a list here of a variety of different disorders ranging from substance use disorders, things like treatment-resistant anxiety, particularly PTSD, some different depressions, including the major depressive disorders and the cancer-related depression, chronic pain, personality disorders, and inflammation. And so there has been a push to see if psilocybin can be useful for these different disorders. But one thing that is not well understood is how this drug could affect embryological development. Oh, sorry. So psilocybin is, as I mentioned, the main psychoactive drug in the psilocybe mushrooms. It is a tryptamine derivative. So you can see here that it undergoes a variety of different pathways to get to the final product of psilocybin. Can you see my arrow when I use it or no? Yes. Okay, thank you. Okay, so you can see psilocybin here, but it is actually metabolized in the liver through first pass metabolism into the metabolite psilocin. And psilocin is actually an active metabolite and is a component of the psychoactive effects that you see after taking shrooms. This drug actually has a high affinity for the serotonin HT2A receptor. And when you look at this drug, it can bind with varying affinities to other serotonin receptors. And kind of, I automatically think of these receptors as being involved in the activity of the brain and in things like mood regulation and in anxiety disorders. However, there are actually additional subtypes that are present throughout the body at which psilocybin seems to be able to function. So if we look here in this figure, what you can see on the left-hand side is a slice through the brain demonstrating the serotonergic pathways. And so if we think about the frontal cortex, for example, and you think about the idea that psilocybin could be used for things like mood disorders, there's some overlap then in the serotonin pathways in the brain and those pathways that might be useful for treating these different disorders. And similarly, if you look here at the striatum and the accumbens, areas that are involved in reward, maybe there's then some overlap of those receptors in how we can treat things like substance use disorders. Now, on the right-hand side, we're actually seeing different serotonin receptors and their presence in the periphery. And so this is a pretty new area for me, thinking about things like this top part here, how activation of serotonin receptors in the periphery could impact how the heart is able to function. And so if we look here, what we can see is that it can change the size of the heart and it can change how the valves and how the heart itself functions, as well as the electrical activity of the heart and the idea that this could be involved in things like arrhythmias. So I'm really expanding out into an area that's new for me by going into the cardiac region. Now, there we go. If we look at the 5-HT2B receptor, one of the things I want to mention is that this receptor is present in the central nervous system. But I want to focus on the idea that this receptor does have both vascular and cardiac effects. The vascular effects include things like pulmonary vasoconstriction, but its cardiac effects, just like I showed you on the last slide, can impact things like both structure and function of the heart. Now, for this series of studies, we actually are using the drug called 1-methylpsilocin. So why are we using that? Well, we're using psilocin because psilocin is an active metabolite of psilocybin. It has a similar half-life to psilocybin, but we add the 1-methylpsilocin here because natural psilocin is very unstable and it's not available in the US. So this is something that would require import from the UK, and it has intensive storage requirements. And Dr. Azine is working on a way to be able to import the psilocin in its natural form, but it is an intensive process. The second reason that we are looking at the 1-methylpsilocin is because the chick embryos that Dr. Azine was using actually don't have the enzyme that's responsible for converting psilocybin to psilocin. And so if they can't convert that enzyme, maybe we're missing some of the effects by that active metabolite. And again, just a reminder that psilocybin is going to be considered an illicit drug requiring approval from the DEA for storage and use, but 1-methylpsilocin is actually not controlled. So it's another reason that we can use this drug effectively for these studies without having to jump through a lot of extra hoops. Now in Dr. Azine's lab, kind of what triggered our interaction with one another is that she was studying the effects of 1-methylpsilocin on embryo development in chick embryos. And so what she did was actually fertilize eggs and incubate them for about 28 hours. Oops, I apologize. For about 28 hours, at which point she treated them with the 1-methylpsilocin or a ringer saline as her control. She then collected the embryos after about 50 hours or 6 to 7 days, and she sectioned those embryos and stained those embryos with a couple of different stains. The first was an antibody stained with they were antibodies stained with PAX7, and PAX7 is going to trace the nuclei and the neurocrest cells. And then we have HNK1, which is expressed in the membranes of migratory neurocrest cells. And so what the point here is that we can then look at how something like psilocin is going to impact the migration of these neurocrest cells into different parts of the chick embryo during development. Now, what she found was that there is a major disruption following 1-methylpsilocin treatment in cardiac neural crest cells and their migration. And so what you're seeing here on the left-hand side in slide A is a control embryo. And here I want to focus on this section right here that we're going to blow up in part C. What you're seeing here is that following treatment with the saline, that these neural crest cells, let me see, are sticking very closely together, and they have a stream that is very narrow. Following treatment with the 1-methylpsilocin, and again, we're going to blow up this section right here in part D, what you can see is that these cells actually have a wider stream, such that they are not traveling in the normal pattern that they would during migration, and they seem to be spreading out instead of sticking tightly together. And so she continued looking at this process of how this migration might impact a phenotype. And so one of the things that she found, and I'm going to point you to this middle piece up here, which should have a little B next to it, and part C. This piece in the middle is treatment with a saline, and this part in part C is treatment with the psilocin. And so what you can see here is that there is an area between the dorsal aorta, which is represented by AO, and the pulmonary artery. And this is what that area would look like under a normal developmental pattern. And that if we look at this developmental pattern, when we apply psilocin, that we actually get defects in those neurocrest derivatives that actually change how that aorta and that pulmonary artery are going to be oriented as the chick continues to develop. And actually, if you look at the chicks themselves, you can see that psilocin significantly changes the development of these embryos as they're exposed to the psilocin. And so not only does it appear to affect things in the heart and those neurocrest cells going to the heart, but also you can see changes in things like eye development, beak development, and shape, as well as some of the overall structures of the body. And so that's something that Dr. Azine is really kind of wanting to understand, is how big and how wide does this effect of psilocin spread throughout the body during embryological development. So her goal, or sorry, to summarize what I've talked about so far, when you look at 1-methylpsilocin, it does seem to disrupt those cardiac neural crest cells and the stream by which they are traveling to their target. And it does seem like if those streams become wider and wider, it will affect how the structures of the heart are actually able to come together and interact with one another. And that was demonstrated by the idea that you have the issues in spacing between the aorta and the pulmonary artery. And so Dr. Azine was really interested then in what is happening here with these neural crest cells. What behaviors are being impacted? Is this simply affecting this specific type of cell, these neural crest cells, or is this generalized to a variety of different cell types? And can we look at some of these ideas with a cell culture model. So she specifically asked me to look using a different cell culture model than specifically a neural cell line. And so I have access to CHO cells here at EKU, and we chose to look at the CHO cells and determine both whether or not the cells remained viable after treatment with psilocin, and whether or not you could see any patterns of morphology changes in those cells. So the first thing that we did was an MTT assay that will measure the cell viability after treatment with the drug. And so we incubated our cells with a variety of concentrations of the 1-methylpsilocin, and we incubated for 24 hours. And we actually looked at this MTT assay and cell viability immediately after drug treatment for 24 hours, and then following 2 days of washout. And so the first thing I want to show you is that if you look at the amount of cell death, or I guess cell viability after treatment with 1-methylpsilocin, for the most part, we saw very little change in any of the cell viability, meaning they actually survived and they were functioning after psilocin treatment. We did have a significant decrease with the 1 micromolar concentration, and this control was the DMSO, which is what the drug was dissolved in. I'm not sure why that 1 micromolar would stand out, since the higher doses are actually not changing cell viability at all. And you can see that this pattern continues, that if you allow 2 days of recovery of these cells, you also see good viability of these cells. They are working, they're functioning. Again, we see with some of these lower doses here, a small increase actually in viability and cell activity, but across these higher doses, there's no change from control. Now, I'll tell you that when we look at cell morphology, these are in the very beginning stages. And so we just literally put some cells onto a plate and added the drug at these different concentrations. And so when we look strictly at these cells in a Petri dish, what we can see is not much in the way of whether or not morphology is actually changing. So again, these are CHO cells exposed to different treatment types, ranging from 0.1 micromolar to 100 micromolar of the 1-methylpsilocin. And I honestly just jumped right in and thought I'd give it a try. And what we're actually going to work on, and I'll talk about in just a second, is how we can actually stain these to actually look at morphological changes and whether or not there are any. So for Dr. Azin's project, she actually thinks that the 5-HT2B receptor is critical in these neural crest cells for the appropriate migration of these neural crest cells to their target during development. And so her goal is to actually use aripiprazole, which is an inverse agonist of the 5-HT2B receptor, to confirm that that receptor is really responsible for the phenotype that she's seeing. She also really wants to look at some of the mechanisms for why this cardiac stream in particular is really showing a big change in big disruption. And so she plans to stain for both caspases and cadherins to look at both whether apoptosis is occurring, whether or not these cells are dying, or whether or not they're coalescing or failing to coalesce, sorry. And then this cadherin is going to really focus then on how these cells are able to interact with one another and stick to each other. Why are they spreading out? What is the problem with adhesion that we're losing when you treat with 1-methylpsilocin? And so in my lab, I really want to dig a little bit deeper with regard to actually characterizing these 5-HT2B pathways as well as the morphology. And so with the CHO cells, they're very hardy. And so I am able to transiently transfect them with the 5-HT2B receptor and repeat the experiments that we just completed, confirming the presence of that receptor with Western blots. I'd also like to vary the timeline of incubation. to see if we can accommodate a little bit more for that half-life. Since it does have a short half-life, maybe if we're looking in the 3-hour time range versus 24-hour time range, we may see some differences. And then obviously I'd like to be able to stain these cells to look at morphology a little bit more carefully and access additional cell lines to determine if these changes are observed in different cell types versus just a CHO cell, and if there's any sort of specificity that way. So I kind of flew through that very quickly. I apologize for talking fast. But again, I wanted to acknowledge Dr. Azine at Loyola Marymount, Dr. Cormier at EKU. In addition to working with a cancer project with her, she allowed me to learn the MTT assay from her, and then Kentucky Embry for the startup funds that were used in this case for supplies to run these experiments. And that's all.
All right, so thank you so much, Dr. Middleton, for a great presentation. We'll open it up for any questions that people have.
And I will try and answer them.
So a question I had, if no one wants to go first, do we know anything about the toxicity of 1-methyl psilocybin or the derivatives or anything like that?
So that was part of the reason that she wanted me to do these MTT assays, was to actually see if maybe some of these neurocrest cells are dying off, or that's the reason they're not able to stick together and travel to their target. And so what it looks like to us from an initial kind of first glance is that it doesn't seem to be killing these cells. They appear to be exactly the same function after psilocin up to really high doses of 100 micromolar as the controls do. So as far as toxicity goes, obviously it's a hallucinogen, so you do have to be very careful if you were giving this to a human, and the hallucinogenic effects, I'm not sure, how much you have to take to make it toxic, but you definitely have to be treating this very, very careful, and what they like to call microdosing.
Right. Any other questions for Dr. Middleton?
Dr. Middleton, that was a great talk. It was really interesting. So with the CHO cells that you looked at, or the data you showed, were those expressing the receptor or not?
Yeah, they're not expressing the receptor. And so we went ahead, I actually did a lot of cannabinoid work, so they are CB1 expressing, but she wanted to just kind of see a separate cell type and see kind of what happens if you put this drug on there. And so is there anything blatantly obvious that's happening? Are we killing them all? And she wanted something that was different than a neural cell line to see how that might be impacted by the drug. And so one of the things I would like to be able to do is, as far as we can tell, these cells do not natively express the 5-HT2B receptor. I haven't fully characterized that. The antibody is on order. But also these cells are super easy to transiently transfect. And so being able to get that receptor in there just to make sure is one of the goals that we have.
Great, thanks.
Yeah.
I just had one question. I'm sorry, go ahead, Alexi.
No, thank you. Yeah, it was very interesting. So I have maybe a more technical question. So when you expose CHO cells, right? So this methyl psilocin, do you know if it's how penetrant this is, right? So essentially how whether that chemical goes inside the cell. Do you know anything about like whether it's able to penetrate the cells very easily, or like whether it's not that, maybe part of that being, not having this effect, right, on the cell culture, but it's just not going inside the cell very easily. What do you think?
Exactly. And so, again, it could be whether or not the cell is actually taking up the drug.
Right.
And so if we think about the 5-HT2B receptor, obviously that's going to be a cell membrane receptor. And so by expressing that or creating a transient transfection, we can actually at least have that receptor on the cell membrane. But looking at how, if this
I see.
actually
So
pass through the cell membrane on its own, that part I don't know the answer to. That's a good question.
got you. Yeah, but you think that, you know, you do need the cell receptor maybe to have an effect, right? So I mean, in other words, the psilocin doesn't need to go inside the cell, right? To
Right.
have, okay, got you. Thank you
Right.
so much. Yeah, very good.
And Dr. Rowe, did you have a question?
I did. This, I guess, is a dosage question because
Yes.
so especially on the chick ones, which you may not know this but for the chick dosage, was the dosage that they were given those chick embryos equivalent to what, say, if a human was taking a typical dose, what might actually be present or being able to be transferred to the fetus, or was it like orders of magnitude more?
Yeah, so very little is known at this point about the transfer of the psilocin or the psilocybin to the fetus. This is really just beginning to be looked at because people are starting to think about using it therapeutically. So this is all pretty brand new. Dr. Azine's dose that she used was the 20 micromolar dose that she was using for her chicks. She's also planning to go back and do a dose response curve if she can. Obviously, doing the chick embryos is a lot more intensive than working with a CHO cell. So she's trying to really kind of hone in on some of her main ideas before jumping into a big, huge dose response study. The 20 micromolar is probably going to be on the higher end. So this would probably be a little bit higher than people would be getting if they were taking psilocybin as a therapeutic, but not necessarily out of the realm of what someone would get if they were taking it recreationally. So that, again, those exact equivalents, I think they're still kind of working that out as far as if it would be, how it would impact the embryo.
Thank you.
Okay, any last lingering questions for Dr. Middleton? All right, if not, thank you so much. And we will switch to our next speaker for the series, and that will be Dr. Laura Rowe. So she is an assistant professor at Eastern Kentucky University in the Department of Chemistry. And she will be talking about the efficacy of antimicrobial peptides as an alternative to traditional antibiotics. So Dr. Rowe, whenever you're ready.
All right. Just give me 1 minute to start my slideshow. Okay, so can everyone hear me and see my screen okay now?
Yes.
Okay, great. So my talk today is about alternatives to traditional antibiotics. So in particular, my Kentucky INBRE funding was for enhancing the efficacy of antimicrobial peptides. And so I'll talk a little bit about that and what those are, and then some other methods of possibly having alternatives to traditional antibiotics. So first, we probably all have some idea of why current antibiotics are limited, and that there's some antimicrobial resistance amongst bacteria. So I want to talk about the issues with that, and then what antimicrobial peptides are, and some methods to increase their efficacy. And then I want to talk about the particular assay that we've been using, and we will continue to be using for testing all these methods. It's called the minimum inhibitory concentration assay. It's a very standard method of testing how well something kills bacteria. And then I focused, especially in this presentation, on kind of a side lane of herbal extractions, and I'll discuss why I've done that and why I'm presenting that as we're going along. All right, so the problem with antibiotic resistance is currently between about 700,000 to 1.27 million people die from drug-resistant infections per year. But this is predicted to be a lot more of a problem in future years. So by 2050, it's estimated that about 10 million deaths per year will be due to antimicrobial resistant infections, which will be more than cancer. So it's increasing for two reasons. One is because of the way that bacteria form antibiotic resistance and some current agricultural practices and things like that. And it's also, the deaths are predicted to increase because there's not very many new antimicrobials coming to market. It's not keeping up pace with how quickly the bugs are mutating, basically. So we probably know basically why this antibiotic resistance superbugs and things like that are developing, right? We've all heard doctors are overprescribing antibiotics when they're not needed. A lot of agricultural practices just use antibiotics willy-nilly, right? Putting it in the cattle feed and things like that. And so, and then the bacteria mutate, right? So the antibiotic kills the vast majority of the bacteria, but there are one or two that have the mutations that allow it to live. Because of the quick replication rate of bacteria, those quickly become the entire population of that particular bacterial population. Then they're all resistant to that antibiotic. And then because of poor water treatment control or agricultural practices, things like that, these antibiotic resistant bacteria just spread all over the place. So there's lots of antibiotic resistant bacteria currently, but there's a lot of different antibiotics too. And the different antibiotics focus on different mechanisms to kill these particular bacteria. So if we look here, right, at different antibiotic targets, we have some that focus on cell membrane, so disrupting the formation of cell membranes. And of course, if you can't have a stable cell membrane for a bacteria, it's not going to live. Whereas others will disturb folate synthesis. So folate is a precursor for the building blocks of DNA, those nucleotides. So if you disturb folate synthesis, there's nothing to build new DNA with, so the bacterial can't replicate. And then there's other antibiotics that target enzymes that are responsible for protein synthesis or nucleic acid synthesis. And so the resistance mechanisms, they can form from the selective evolution, right? From the pressure being this antibiotic that has been imparted into the system, all these different ways. So you can have the bacteria has one or more amino acid mutations that affects whatever that antibiotic was targeting, right? Something in the cell wall or the protein that has to do with the DNA or RNA synthesis, or the protein that has to do with the translational machinery, right? If there's a single or multiple amino acid substitution in one of those enzymes, then that antibiotic may no longer be able to bind to that. And so it gets away from the antibiotic cell death. There's biosynthesis bypasses, so coming up with different ways to synthesize folate, for example. And then there's these other methods. The certain bacteria have evolved enzymes that specifically will metabolize these antibiotics, right? Or they've evolved efflux pumps that will specifically chuck out these antibiotics as soon as they come in. So for all the different antibiotics we have, the bacteria evolve new methods of getting around them. So if you look at ways of reducing this antimicrobial resistance, there's lots of ways, right? We've got this nice chart. I like to steal charts and graphs and figures from Google and then cite them down here. But I'm going to focus on this one, the research and development of new therapeutics. So a problem has been, if you look from 1980 to 2014, I don't have the data from 2014 to 2022, but it's not much more than this. I had like from 2012 to 2022, it was 17. So it might be like 8 or 10 since then. But the number of antibacterial new drugs that have been approved has been decreasing overall. And the reason for that, from the articles that I've read, is largely because pharmaceutical companies aren't making enough money off of these. So a good antibiotic or antibacterial is kind of a one and done thing, right? You get an antibiotic infection, you take antibiotic, or you get a microbial infection, you take an antibiotic for a couple of weeks and it's done, right? You don't have to take that antibiotic anymore. Whereas a lot of the biological medicines are coming up with, such as biopharmaceuticals for chronic diseases like autoimmune diseases and diabetes. These are chronic diseases. You have to keep taking that medicine forever. And so they're a lot more profitable. So a lot of pharmaceutical companies are not putting a lot of time and money into developing these because they don't see it as a money-making proposition. So basically, both of these aspects are contributing to this problem that's going to have a lot more deaths in about 30 years, right? Both the resistance of the bacteria and that we're not developing enough new therapeutics. All right, so enough about why this matters. So what are antimicrobial peptides? So that's kind of self-explanatory, the name of them, right? I'm going to call them AMPs from now on. They're relatively short sequences of amino acids that, in my case, I'm looking at that kill bacteria, right? So this here is an overall structure of amino acid. We have the ball and chain formation where each of these represent different amino acids. So they're found naturally in lots of different organisms, including humans, right? Humans have defensins, which is something that we secrete on our skin to help us keep bacterial infections on our skin away. So you can have just the regular amino acids in these antimicrobial peptides, but you can also have a lot of what's called post-translational modifications. So, and the organisms that produce these on their own do this in their own cells, right? So you can have these AMPs decorated with sugar groups, lipid groups, or unusual amino acids that have been post-translationally modified from the standard 20, right? They can also be cyclized. And so this figure here is just showing, this is the shorthand for glycans or sugar groups. So a lot of, for example, vancomycin, which is an FDA-approved antimicrobial peptide, but it's called a glycopeptide because, and it's a very important one, because if you have a antibiotic resistant infection and they've tried several antibiotics for you to try to get rid of it, and none of them have worked, kind of their last line of defense, or one of their last lines of defense, is vancomycin, which is an AMP, but it's a glyco antimicrobial peptide. So it has some sugars attached to it, and it has a bunch of post-translational modifications and things like that, but it works well. So they haven't decided on how to classify these. There's various ways. You can classify them based on what organism they come from, right? Or you can classify them based on what is the most prevalent amino acid in it. Or you can classify them based on their secondary structure. Or you can classify them on their activity. So if you look over here on the activity, you can see there's a lot of antimicrobial peptides that do a lot more than just kill bacteria. So this one, right, the one with the big, that's the only one that I'm looking at. But they have some that have anti-cancer activity, some antifungal, antiviral activity. If you look here in this pie chart, right, about of the known antimicrobial peptides, about 60% of them are antibacterial, have antibacterial effects. But there's other ones, but I'm just going to be talking about the antibacterial ones. All right, so if you're talking about antibacterial AMPs, there's different modes of action. Just like antibiotics kill the bacteria in different ways, these AMPs kill bacteria in different ways. In the sake of time, I'm not going to go through all of these ways. I'm just going to focus on the one that is circled in red, because the type of AMPs I'm looking at are the ones that disrupt membrane formation, so cell membrane formation. And these are preferred in many cases because compared to metabolic disruptions, they think it's less likely for bacteria, or it's harder for bacteria to evolve antibiotic resistance to these. Because if you're blowing up the cell wall, it can't reproduce at all, right? You can't go through the motions where mutations might happen that introduce novel mutations that make it circumvent the AMP's toxicity. Whereas if it's a metabolic disruption, they have a little more time to possibly have these mutations occur. So cationic alpha helical AMPs are the one, this kind of my model AMP that I'm looking at, or we're looking at, my research group is looking at. So this is alpha helices, and meganin is a AMP that has been isolated from frogs, from the skin of frogs. So if you ever wonder how frogs can sit there in nasty swamp water all day long and just be fine, it's because their slimy skin is covered with meganin. all right, and other types of AMPs that keep any nasty bacteria and stuff from infecting their skin. All right, so, but how these work, one, they're membrane disruptors, and they work by having different types of amino acids on the different sides of the alpha helices. So for example, on one side, they will have charged amino acids, lysine, for example, that have a positive charge. And on the other side, you'll have hydrophobic residues, phenylalanines, tryptophans, things like that. And so what this sets up, one, it sets them up to much selectively prefer bacterial membranes, because bacterial membranes have this negative charge, right? So that positive charge is going to be attracted to that negative charge, whereas eukaryotic, such as mammalian cell membranes, they're overall neutral charge. So it sets them up to preferably go towards the bacterial cell membranes. And when they attach there, eventually these hydrophobic residues are going to kind of wiggle in to the hydrophobic area of the cell membrane. So you have the hydrophobic region of this lipid bilayer there, and they're going to kind of wiggle in and want to associate in with those hydrophobic groups. All right, but then you have these, if two of them have kind of wiggled in there, those are both positively charged on the other side. And of course, that's going to be repelled. All right, so those positive charges are going to repel and basically punch a hole into this bacterial membrane. So if you punch enough holes into the bacterial membrane, it's a dead bug. So just to review the difference between gram-negative and gram-positive bacteria, because the results I'm going to show is from a gram-negative bacteria called E. coli. Now we have some gram-positive bacteria that we're going to test soon, but we just haven't had a chance to do that. So gram-negative bacteria have this double layer, right? This double cell membrane. And this causes them to be more difficult to kill in many cases than gram-positive bacteria. So we kind of tested the harder one to kill first, and then we'll test the easier one to kill, just because it's easier for molecules to get into the gram-positive bacteria in general, because they only have to bypass this one cell membrane and not both of them, or this one lipid bilayer. So again, to kind of hone in on the differences in these cell membranes, both gram-positive and gram-negative bacteria, the outside of the cell membranes have an overall negative charge. They have the overall negative charge for different reasons, but it's still an overall negative charge, which those cationic AMPs, right, are going to be attracted to, much more so than the neutrally charged lipids associated with a healthy mammalian cell. All right, so Maganin 2 is the one that I'm kind of using as my model. And the reason I'm using it as my model is, one, it's well studied. All right, we know what it's made of, we know how it works, we know why it works, and also, and not insignificantly, it's commercially available. All right, so I can just buy Maganin 2, and if I want to attach certain groups to it to increase its targeting capacity or to increase its toxicity, right, I can just buy commercially available Maganin 2 and then use some chemical methods to conjugate something to the end of it. Or also, it's relatively small. All right, 20-something amino acids. And importantly, there's no complex post-translational modifications. So vancomycin is a wonderful AMP, but it's got tons of crazy post-translational modifications that you have to pretty much grow it in a cell, right? You have to grow it in a cell that's specially designed and then purify it from that cell. Well, that's a lot of work, right? So I want something that has virtually no ideally no post-translational modifications. So it's cheaper to buy. And also, if I want to use solid phase peptide synthesis to make these, or say put in an unnatural amino acid, right? So if I wanted to change this lysine to a fluorinated lysine, which you can easily do using solid phase peptide synthesis. And I can do that because I don't have to worry about a bunch of post-translational modifications on this peptide after it's synthesized. But again, it has the two different surfaces, right? It has here one side that has the positively charged lysines, and then it has the other side that has the hydrophobic groups. And these two are basically showing the same thing. This is just telling you the exact identity of all the different hydrophobic groups and the exact identity and positions of all the different lysine groups. So Maganin 2, like I mentioned, came from a frog. It's been shown by many different researchers to have not only antibacterial activity, but also antibiofilm activity, and it's also stable in salts. All right, so here's the issue, why I've kind of selected to show you results that are maybe not the results I want to show you. And this is a patent issue. So if any of you all are planning on have some intellectual property that you think might be worth something eventually and plan on patenting it, you have to think about when your public disclosure comes out. And I learned this from talking to Daniel Scott at Center College, who went through this, and also from talking to the Kentucky Commercialization Ventures. So I went and talked to them earlier this year. So for EKU, if you have intellectual property you're thinking about patenting, the first thing is you've got to submit your invention disclosure to Eastern. And then Eastern and quite a few other regional Kentucky universities, they've partnered with Kentucky Commercialization Ventures to determine if they want to pursue this patent, if they think it's worthwhile. And they've got 90 days to decide whether or not to do it. If they do decide they want to patent it, then they will pay for all the patent application procedures, but it takes 2 to 6 months to get all that provisional patent stuff done. But if they decide not to patent it, then they got to tell you within 90 days, so then you can go ahead and patent it if you want to. But of course, you have to pay for it if you patent it yourself, but you also get 100% ownership of that patent. Now, the problem here is it is highly recommended by multiple people that you don't publicly disclose, which means give a presentation on the specifics of your intellectual property until you've submitted this disclosure to your university, because the time this takes, right? 90 days, 2 to 6 months. And the federal law is you have 1 year grace period to submit your patent application after you've publicly disclosed it, which means after you've given a presentation. So I have not submitted my discovery disclosure to EKU because I haven't had no time, all right, to do that. So I'm planning on doing that this summer. My last final was yesterday, but I haven't done it yet. So I'm not going to publicly disclose all this stuff yet, is basically the name of the game. And I actually had to think about this, let's not read out their faces, when I was thinking about projects amenable to my students. Because one, I wanted my students to present their research at the ACS conference this spring in Indianapolis, which they did. This is Jessica and this is Hannah. They presented some research like I said, at the ACS conference, but also both of them took research as a course. So in chemistry, you can take it as a course, and at the end of the semester, you're required to give a public presentation, right? Or semi-public presentation. So I had to think of things that they could present before I've submitted this. So that's what brings us up. So I'm going to tell you some general methods for AMP stability, and I'm going to tell you what we actually did, and then we'll probably be out of time. All right, so some general ways this is to increase stability of all proteins, right? Not just antimicrobial peptides. So some general methods is cyclization. So you can, a linear peptide, you can cyclize it using various chemical methods or biological methods called enteins. And this cyclized peptide is much more stable to protein degradation in living fluid. All right, it doesn't degrade nearly as fast. Also, and this has been done in many other proteins, so this is public knowledge already, you can often add a fluorine group to amino acids in order to increase the stability of the protein. So exactly why fluorine tends to increase stability of proteins is not exactly known. They think it might increase the stability of the hydrophobic core of the protein if it's a globular type protein. But this has been done. And in fact, fluorine increases the biostability of a lot of pharmaceuticals that aren't protein based. So I think it's like over 30% of small molecule pharmaceuticals, they've chucked a fluorine in there specifically because it increases stability. So that means that the drug doesn't degrade when you take it, right, as quickly. Another way is using what are called beta amino acids. So instead of alpha amino acids, incorporating these beta amino acids can often increase stability because the cells don't recognize, your enzymes that go around degrading proteins don't recognize them as quickly. All right, so they have a longer half-life in living fluid. And a general method for increasing the efficacy of AMPs, I'll just focus on what's called antibody drug conjugates, which are a big field in biopharmaceuticals right now. Antibody drug conjugates. I'm not going to write all that down. But the general method that they use could also be used for AMPs. So here you have cancer cells, which many times cancer cells have specific receptors on the outside of them or a specific shape. So antibody drug conjugates have an antibody, and this end of the antibody selectively recognizes cancer cells. So that increases its selectivity, right? Instead of going around and hanging out next to all cells, it hangs out next to cancer cells because this Fab region of the antibody, right, the antigen binding site, recognizes cancer cells. So that increases the selectivity. And they can also attach a toxin, right? They attach for radiation, for more advanced radiation therapy in cancer now, they'll attach the radioactive compound to that antibody, so it targets it to the cancer cells instead of all over the place. They can also have like a cleavable linker there, that because if around a certain cell type, the pH or something is slightly different and the toxin will cleave and basically be released as a toxin at that time. So these are general methods. Now, I'm not gonna show you any of that. I'm gonna show you something related. So this is culinary and medicinal herbs. So when I was thinking of data that students could, one, that students could present quickly, and two, that wouldn't cost me thousands and thousands of dollars of AMPs. So when I have students training for this, for these MIC assays, they have to use a lot of the antimicrobial, whatever it is, to do these MIC assays. And some of these, Meganin is like $1,000 for 10 milligrams. So when they're first learning how to do this stuff, and they have to learn how to do these MIC assays and serial dilutions and all of that, I don't want them using the antimicrobial peptides. So I thought, well, let's look at some common herbs, because there's a big push for basically making cheaper home remedies, right? So you can have culinary and medicinal herbs and see if these have any antimicrobial effects using the exact same methods we'll use with the antimicrobial peptides, kind of just to explore this and have some data that they can present and to have them practice with these methods before we chuck the $1,000 every 10 milligrams AMPs at them. All right, so that's what we did. We extracted all of these herbs and tested them. Herbs, however you want to call it. So we mainly use culinary herbs. A few of these aren't, right? Burdock root, elderberry. If you ever had burdock root tea and elderberry tea, it tastes awful. So those are definitely not culinary, they're medicinal herbs. But mainly I chose these because these are what I had in my cabinet, right? They're what I had. So that's what we used. I had some CBD oil because who doesn't? And so we used some CBD oil as well. Genetian root, angelica root, those are definitely more medicinal. All right, so we extracted these using 3 different methods and then tested them using an MIC assay. So the 3 methods of extraction, one was a water extraction, which is going to extract polar molecules from the plant. The other was an ethanol extraction, which is dual polar, so it will likely extract both polar and nonpolar molecules. And the third was a canola oil extraction, which is mainly going to pull out nonpolar molecules from those herbs. So the whole process for this MIC assay, which we did for all of these herbs, goes like this. We focused on E. coli. All right, so you have to do these for each individual bacteria. We focused on E. coli because it's a biosafety level 1. It's very easy to work with. So you streak a plate with them, you make a fresh inoculum, and then you prepare, while this is basically growing, you prepare different dilutions of your antimicrobial agent. In this case, it was our herbal extractions, however we extracted them with the different methods. You inoculate these on 96 well plates, and then you measure the absorbance after a certain amount of time. All right, so you put the antimicrobial agents plus, right, some of your culture, and then you see what grows. So you absorb absorbance at 600 or 630 nanometers, it's turbidity basically, and you measure this against the turbidity standard. And then you see the ones that have the highest absorbance at this particular wavelength have the most bacteria in them, because that's what's causing the absorbance. So, and then we plotted these absorbance values and compared them with regular antibiotics. So here's some results. We did everything in triplicate. So these are a couple regular antibiotics that were basically our control values. All right, so we have our two controls on all experiments, which are important. We have our growth control and our sterile control. So our sterile control contains everything except no bacteria. All right, we don't put any of that inoculum that has the E. coli. There's no bacteria. So that should have a very, very low absorbance, right? And then the growth control is the one that has everything except the antimicrobial agent. So that's it, how the bacteria would grow if nothing's added to it, nothing to inhibit its growth. So you can see for kanamycin and chloramphenicol that there is a lot of antimicrobial effect, right? Until you get to really low concentrations, right? When you get down here, it starts chuffing off, all right? And then we had a few really good hits for our herbs, and these were all our water extractions. So the only data I'm going to show in the interest of time is the water-based extractions. So sage worked pretty well, right? You needed a higher concentration than you did for the normal antibiotics, but it still did pretty well. Dried mint also worked pretty well to inhibit bacterial growth. A couple other good ones, garlic powder worked pretty good. And surprisingly, dried eucalyptus also worked pretty good. Again, it had to be higher concentrations than the antibiotic itself. Now to summarize all these, so we don't go through all of these graphs, here's the list of all the ones we did. Water extracted against E. coli gram-negative bacteria. So the ones with the red star had a strong antimicrobial effect. And that was sage, thyme, dried mint, basil, garlic powder, and eucalyptus. It wasn't as good as the regular antibiotics, but it still was obviously an antimicrobial effect. Now the green ones had a mild antimicrobial effect. So it was less than the growth control, but it didn't really bottom out like the sterile control. Right, so those were allspice, lavender, angelica root, red pepper, sarsaparilla tea, and coffee. And if there's no other marks, then they had no antimicrobial effect. Now, some of you might be looking at this and being like, but I've always heard clove kills is very antibacterial. Yes, there's a lot of literature research saying that clove and lots of these other ones have antibacterial properties, but those were almost always essential oils. So those were oil extractions, which suggests that the antibacterial molecules in those are nonpolar, all right? And we wouldn't have got hardly any of those out with our water extractions. So future work is ethanol and oil extractions. They've already been done and tested. We've actually done the MIC testing, but I haven't got all the data crunched. So my students crunched a lot of the data, but I haven't looked at the controls yet. So I need to look at the controls and compare and get the error bars and things like that, make sure all that looks okay before I present it. I suspect from the data we have so far, more herbs show antimicrobial activity from the ethanol and oil extractions than from the water. We need to standardize our extraction time and temperature. So the extraction time, the incubation time before we use these solutions varied from 3 weeks to 4 months, all right? And so we need to standardize that. We also need to test gram-positive bacteria. And what we've kind of started on, but probably won't get to for a while, because we got to wait for these extractions to sit for a few months, is extract Appalachian medicine specific herbs. So such as yellow root, boneset, goldenrod, etc., that are kind of Appalachian traditional folk medicine. And then how we are going to use this with our antimicrobial peptides is to see if these herbs are adjuvants. So adjuvants just mean things that make other things work better, right? So if these could be, if you added these to an antibiotic, does it make the antibiotic work better? Or if you added these herbal extractions to an antimicrobial peptide, does it make the antimicrobial peptide work better or not, right? So we could, for example, test these herbs with vancomycin, which is an antimicrobial peptide that is used in humans, and see if we add some of these with the vancomycin, does it make it work better or not? So I think I have one last slide before acknowledgements. So I really like the idea, actually, which I wasn't even thinking about this last year. I like the idea of exploring Appalachian folk medicine because it's largely unexplored. There's a huge amount of scientific literature done on Chinese traditional medicine, but there's almost none done on Appalachian folk medicine. However, Appalachia has one of the world's most ecologically diverse temperate forests. We have many old growth forests, and Appalachian medicine combines is a combination of Native American medicine and European herbal medicine. So it's kind of unique, and it combines those two traditions. And also, I think students are very interested in this. It very much seems to increase the engagement of the students when I talk about this, because they think, oh yeah, that weird oil my grandma gave me that helped with this. Maybe I could study that scientifically and really engage them in science as a process that they can do in their own community, and not some magical thing that they do far off in the big cities. So with that, I will go with my acknowledgements. Mainly my students, all right, Jessica Boggs, Hannah Leroy did most of this work this year. Edward Sackall did some as well. I had other students who worked on other projects last year, Churro Taylor, Adam Bradley, and Emily Scheib. They've all done a really great job. I think these MIC assays are especially good for undergraduate researchers, because after about 3 weeks of training, they can really start producing useful data with very little supervision. So I think that's really important for undergraduates. So I really like this, because these screens, after about 3 or 4 weeks, they can do them on their own. And of course, my funding sources, Kentucky INBRE and NIH for a startup grant last year, and for an idea grant this year, Eastern Kentucky University for supporting me and giving me some course release and things like that, and Kentucky Commercialization Ventures for giving me advice about patent stuff. And thank you for listening. And that's all I've got.
All right. So thank you so much, Dr. Rowe, for that wonderful talk. So we will open it up for any questions.
Hey, yeah, it's very interesting. Yeah, I really enjoyed that. In terms of this microbial peptide that you talked about, this Magainian, right?
Yes.
So I noticed that it has string of lysines, and this is what you talked about as well. And also on the other end, right, this hydrophobic side is made of phenylalanines.
Yes.
Do you know if you change those lysines to, say, arginines, would it increase or decrease the effect of this AMP on its antibacterial properties? And also, if you change, say, phenylalanine to another hydrophobic amino acid.
Really, I need to do a literature search, because I would think that something like that has been done before, but I don't know of the study off the top of my head. Of course, if it hasn't been done, then that's number one thing we should do. I do know of studies for these alpha helical peptides, but I don't know if it applies exactly to Magainin 2, in that if you increase the hydrophobicity on that one side, you increase its capacity to kill bacteria. So let me go back. Go way back. All right. So I don't know if it's been on Magainin, but on these general alpha helices, I do know that if you increase the number of hydrophobic residues on this side, there have been some studies showing that increases their ability to kill bacteria. But yeah, I don't know if changing this to tyrosine or tryptophan, I don't think tyrosine would be good, because they have that OH group sticking out. And tryptophan, maybe, but it's not as hydrophobic as phenylalanine, because it has those nitrogens, right? So I don't know, but I'd have to look in the literature to see if that's been done. But there has been, if you add a few more of these phenylalanines, right, that it will increase its ability to kill bacteria, but it's a balancing act, right? If you get too many of these hydrophobic residues over here, they might all clump together in like what they call an inclusion protein, which is basically a big glob where all the hydrophobic residues clump together in the center to get away from the aqueous environment. So there's definitely a balancing act.
So yeah, so you think that this is just enough of those phenylalanines to prevent aggregation, right?
I can't say that for sure. I haven't done a thorough enough literature search on Magainin to say that.
Right.
But I think it's a possibility.
Great. And also, just like a general question, do you know if plants have those AMPs as well?
Oh, let me look at my chart. Yes. According to my figure, they do.
Oh, yes. Great. Thank you.
You're welcome.
When in doubt, always go back to the chart. All
That's right.
Any other questions for Dr. Rowe? All right. Well, if not, I want to definitely thank our speakers, Dr. Lisa Middleton, Dr. Laura Rowe from Eastern Kentucky University for sharing their research with us. I'd like to thank all of you for attending, and I hope that you have a wonderful afternoon. All right. Bye.